Hello, everyone. Welcome to our academic success workshop for biology video. I'm Shreyas,
And I'm Noah.
and we're just going to cover some bio topics in this video so we can get started. So what is biology, you might be asking? Biology is the study of life and living organisms. This can range from large-scale biology, such as animals and plants, to small-scale biology, like organelles and cells and stuff like that. And then we will cover our first topic now, which will be.
Yes, we're going to go over some of the cell parts. Next slide, please. So what is a cell? Cells are the fundamental unit of life. All organisms on Earth contain cells, and this is from plants to animals. Organisms can be unicellular or multicellular, which means that some organisms have a lot more cells than others. And then multicellular organisms have different cell types for different functions. So this is kind of shown in this diagram down here, where a neuron is a different type of cell than maybe a liver cell. So first we're going to ask, what is the difference between an animal and a plant cell? So they do contain many of the same aspects in organelles, which are the tiny little organs that are in cells, except the plant cells has the extra organelles of cell walls and chloroplasts. And I'm going to expand upon this a little later when we go over all the functions of some of the main organelles in a cell. Okay, so first here are some of the organelles that both plants and animals share. So first we have a cell membrane. This is the outer proportion of the cell. It is made up of a phospholipid bilayer, and this contains other proteins which allow for cell functions and for molecules to travel in and out of the cell. Then we have the nucleus. This is the brains of the cell, and it also contains most of the DNA for the organism, as well as this is where protein synthesis begins for protein synthesis. Then we have mitochondria. This is kind of like known as the powerhouse of the cell, as it generates energy for the cell by creating ATP, but it also regulates homeostasis and produces steroids, which is some other functions that not a lot of people know about. Then we have ribosomes. These are found in the rough endoplasmic reticulum and throughout the cell. This is where proteins are actually made, and then these proteins are later carry out cell functions. Then we have the Golgi body, and this prepares molecules for transportation in and out of the cell. And finally, we have the cytoplasm. So this is kind of like the gel-like fluid inside the cell. It surrounds the organelles and allows for the cells to have space for chemical interactions. And these are two organelles that are only found in plant cells. So first we have the cell wall. So as it says here, it's only in plant cells, and it provides extra strength and stability and for water balance. So plant cells need these tougher cell walls in order to have enough strength to kind of hold themselves together, because unlike animal cells, they don't have like bones. So the cells need to be a little bit stronger. And also they have chloroplasts. So these are also only in plant cells. And this is where photosynthesis takes place, and it turns light into energy, and is also what makes plants green.
Very cool. Okay, now we will cover our second topic, cell division. So cell division, you might be asking, the term for that is mitosis, and it's used for all somatic cells. This is where one parent cell turns into two daughter cells. For gametes, like sperm and eggs, they undergo the process of meiosis, which is a different topic, which we will not be covering in this video. But there are four main stages of cell division. The first one is interphase, or this is like the first step. It's like the resting step for mitosis, in the sense that when the cell is not undergoing cell division in the moment, it's an interphase, and that has multiple steps to that as well. But for the actual first step of cell division, we will look at prophase. So prophase is the initial step for mitosis. It's where the chromosomes condense and the nuclear membrane disintegrates on the outside. And then the centrosomes move to opposite sides to form the spindle fibers, which will align the chromosomes in the next step, as you'll see. So we have metaphase, which is the second stage of mitosis. This is where the spindle fibers will attach to the centromeres of the chromosomes. And then this is what causes the chromosomes to line up in the middle. And a helpful hint, I think, with this is that when you see the word metaphase, you can think meta, middle, because that's what the main purpose of metaphase is. Then we have anaphase. This is where the centromeres split apart, causing sister chromatids to separate. And the sister chromatids are pulled to opposite sides of the cells by the spindle fibers. This is the step that makes sure that both daughter cells has the correct amount of chromosomes and DNA, and that they're going to be equal on both sides. And a helpful hint for anaphase, I believe, is ana away, A and A. Just think that it helps people remember that anaphase is when the spindle fibers move or the chromatids move away from each other. Then we have telophase. This is where the identical chromosomes located at each opposite pole of the cell you can see like the chromosomes go into the separate nucleuses, and the nuclear membrane begins to form around each set of daughter chromosomes. And that goes along with cytokinesis, and this is kind of where the cells will begin to separate by creating this cleavage furrow. And this is the step where that one parent cell has now turned into two daughter cells, effectively creating two identical cells. And then this is a diagram that highlights all of the stages of cell division. And then finally, we will now cover genetics as our last topic in this video.
So what is genetics? Genetics is the study of hereditary or the passing of traits from parents to offspring. And this is the reason why children tend to look like their parents, because their traits are getting passed down. Gregor Mendel is the father of modern genetics, and he discovered the fundamental laws of inheritance. And he was actually like a late 1800s biologist. And he did this by tracking inheritance through research on pea plants.
So when we talk about basic Mendelian genetics, as Noah said, it's the traits are passed down from parents to offspring. And in the most basic sense of doing a Punnett square, each parent has two independent traits that can be passed down, and then these can create different combinations in the offspring. So if you look at this diagram, we have a green pea plant, which has two lowercase y's, and a yellow pea plant, which has an uppercase y and a lowercase y. And then you can kind of see how they go together in each box, and where the two small green ones create a homozygous recessive trait, the green, while the yellow will create a heterozygote and have the yellow phenotype. And then Noah will go into that in more detail.
All right, so inheritance. So see, these are kind of the ways that inheritance is passed down. So first we have homozygous. So the traits are the same, and examples are. So looking at the example to the right, our example here would be two capital B's and two lower B's. These are homozygous because it's the same one. Then we have the opposite of that, which is heterozygous. So the traits are different. So once again, we have in the top right box and the bottom left box, we have a capital and a lowercase b. Obviously, these are two different fonts of the B, so they are heterozygous. Then we have the dominant trait. So these dominant traits are always expressed even with one copy of the trait. As you can see with the example again, the dominant trait is always expressed even if there is the recessive trait. So for instance, in the I one, the dominant trait is the uppercase B, which produces a brown eye. So even though in two of the boxes, there's an uppercase and lowercase b, because it has the uppercase B, it will always be expressed. So the brown eyes will be expressed and not the blue eyes. Then there's the recessive trait where it needs both pairs of the trait to be expressed. So once again, for this example, you can see there's two lowercase b's that makes the blue eye, but it needs to have both two lowercase b's. If there was a lowercase and an uppercase b, it would once again produce the dominant brown eyes.
So finally, to actually solve the Punnett square, as I was kind of explaining before, you kind of place the parent on top and on the left side of the box, and you kind of just organize them by putting each letter into that box. And so in this case, the red big R and the green big R will come together and create an offspring that has both a big red and a big green, and same process. And this can help you determine the percentages or the probability of the offspring having a certain phenotype from their parents. And that is it for the video. Thank you for watching, and hope you have a great day.